Okay, good morning, everyone. Today's music selection, by the way, is music I listened to in college a long time ago when it was rainy and crappy outside. First off, so with that, I would like to introduce Mike Meehan, who has an announcement about those wonderful iClickers. That was sarcastic.
Okay, I'm sure you received several emails last night about the iClickers. I've received about 75 emails back in response from students. Okay, first off, last name is your last name, UC. First name you're using at UC. Your legal names, not made-up names, not nicknames, your actual names. The remote ID is the ID that's on the clicker. It's available on the back. If you don't see it there, it's then in the battery compartment. If you still can't find it, I've already had several office hours where you can come to my office, send out a signal, I'll tell you the ID. If you've not yet done that, which you should have, I will again do that today from 1 to 3, the last time. Under the remote ID, there are no letter zeros in the remote ID. If you see something that looks like an O, it's the number 0. No O's in the remote IDs. Now I've got emails, but my last name has an O in it. That's okay. It's fine to have your name have O's, okay? It's the remote ID that can't have O's, only zeros. Any questions about that? You can re-register your remote ID. That is the solution. If you're not sure you registered it, just re-register it. I've gotten like 50 emails from students saying, can I just go back and check for them? You can check for yourselves. It's easy enough to do. The only exception is if you lost your iClicker during the process and you got a second iClicker, then you may not want to register the new one because since you lost it, you won't remember that ID. So if for some reason, and I've gotten some emails from students who have used 2 iClickers because they lost one, then come see me, email me. That's when we definitely need to work something out. Any questions about the iClicker stuff? I just am amazed how it worked. Mike, yes, right, question. Okay, the question is, will the registration still go through if you put it as an O? I don't know. I don't operate the program. What they should, the problem is that your remote ID can have alphanumeric combinations. So I don't think they've programmed it so that if it ever sees the letter O, to reject it. So that's for the remote ID, okay? So if you went and put it in as O's instead of zeros, just again, re-register it, okay? There's no harm in re-registering. Just go to the website again and re-register. Any other questions about the clicker? Again, all right, thanks. Sorry for all the announcements. This is the end of it, okay?
Okay, thank you, Mike O.
Thanks, Ariel.
Wait, my name has a Y, not an O. Oh, wait, Firestone. That's right, there's an O in there. 0. Wait, aren't the O's the fatter ones, right? Okay. Thanks. Herein lies why I use this as my technology. It always works. Piece of chalk. Hope this is still working here. So today we're going to do spermatogenesis. We can leave that up for a few more minutes if you like. Today we're going to do spermatogenesis, male hormones, oogenesis, ovum development, menstrual cycle, or if we get that far, female hormones. So a little bit of a review. You should know the difference between type 1 and type 2 diabetes as a way of what it does is you regulate the insulin receptor signaling pathway, binding, dimers, activation of tyrosine kinase, phosphorylation of substrates, phosphorylation receptor, binding of signaling molecules. And if you have a defect at any point in that pathway, you can end up with a disease state. And that's mostly how type 2 diabetes arises. Over the years, as you get older, you don't respond as well, not so much with insulin production, but with the response to insulin. And many times it's not the receptor, it's downstream signaling events from the receptor. And eventually it turns out that you then, type 2 diabetes manifests itself. And those of you getting into this as a field, you'll see there's many, many reasons for it, all related to receptor signaling. Okay? Then we talked about glucagon receptors. And these are for those of you who did not eat breakfast this morning. The idea is to bring glucose into the blood. In the inactive state, this receptor is a 7-pass transmembrane receptor bound to a G protein. The G protein has GDP bound to it. This is inactive. Hormone binds. The on switch is a GDP, GTP exchange, which we talked about. This GTP-bound G protein activates adenylate cyclase to produce cyclic AMP. The off switch, the G protein itself is an enzyme, has a GTPase. And I put on the board there just a loose structure, very loose structure, where you cleave off that gamma phosphate group. That happens while it's attached. The G protein in the GDP-bound state now has a higher affinity for the receptor, rebinds the receptor into the active state, and it stays there until it sees a hormone. So on switch exchange, GTP, GDP exchange, off switch, GTPase. Everybody see that? Questions? Cyclic AMP initiates the response, activates protein kinase A. Protein kinase A is a serine threonine protein kinase. It starts out in an inactive state. Cyclic AMP binds to the regulatory subunit, uncouples the two subunits. The catalytic subunit then runs amok in the cell, has many, many substrates. In the cytoplasm, it converts glycogen into glucose, therefore produces glucose. Those of you who had a nice big meal last night, that's what's going on when you don't eat breakfast. It also goes into the nucleus, phosphorylates a specific transcription factor, kicks on the synthesis of gluconeogenic genes from the name, producing new glucose molecules. Okay? So together, you produce more glucose, secrete it out into your blood, and off you go. This is when you're hungry. Not starvation, but this is hunger. Okay? As we talked about, regulating cyclic AMP is incredibly important. Cholera toxin keeps the G protein in the GTP-bound state. Therefore, it keeps adenylate cyclase on. Therefore, produces cyclic AMP at very high levels. Not good for you. Cholera is obviously a very disastrous disease. In addition, you can regulate cyclic AMP levels by phosphodiesterase. So it converts it to 5' AMP. You lose the response. Those of you who had coffee this morning or tea or ate chocolate, not too many people eat chocolate in the morning, but there are a few. All those have caffeine or a derivative of caffeine that inhibit that phosphodiesterase. Okay? So someone who has cholera, for example, the last thing you want to tell that person is drink some coffee, because you have adenylate cyclase on and you kick off the ability to turn over cyclic AMP. Okay? You give them clean water, mostly. There's other things you do, but you certainly put them on clean water. Okay? Any questions? Okay, so know the difference between when you're hungry and you have glucagon pathway and when those of you who had breakfast have the insulin pathway. Then we got into spermatogenesis, which we'll continue with. It is in the males, of course, it's a continuous process because of negative feedback. We'll talk about the hormones in a few minutes. It all takes place in the seminiferous tubules, which are in the testes. The three kinds of cells are the spermatogenic cells, of which there are several. We'll talk about right now in a minute or two. You have the Leydig cells, which convert cholesterol into testosterone, and the Sertoli cells that convert testosterone into dihydrotestosterone. When we talk about the negative feedback system, you'll see how it all kind of comes together. Okay? And where we left off was looking at this is the cross-section of the seminiferous tubules. This is the basement membrane. Leydig cells are outside. Sertoli cells are inside. The Sertoli cells kind of contact each other. And where we left off was it forms a Sertoli bridge. And what happens on this side of the Sertoli bridge with the spermatogenic cells is different as to what happens on the inside of the Sertoli bridge. And it's very important to see this. So this is a magnification of that. So where we ended with is you have cells that are continually dividing called spermatogonia. And they divide by mitosis. These are true diploids in that they have 46 chromosomes and 2 cDNA content. Remember, the object of this pathway is to get a haploid gamete, right? What's the DNA content of a haploid? 23 chromosomes and cDNA. So half the DNA, half the chromosomes. But it does it in a slightly different way. What happens is, it's not really clear exactly which spermatogonia undergo this step, but the next step in the process is the meiotic duplication of the DNA. And you get a primary spermatocyte. Okay? And the primary spermatocyte is 46 chromosomes and 4 cDNA. So the primary spermatocyte then goes through the Sertoli bridge, and here's the rest of the Sertoli bridge. So this board is now this inside part of this lower magnification of a cross section. So the primary spermatocyte, the one that undergoes the meiotic duplication with the extra DNA, goes through the Sertoli bridge, Sertoli cell bridge. And then there's a completion of meiosis. So then you have the first meiotic division, and you get secondary spermatocytes. And the secondary spermatocytes are 23 chromosomes and 2 cDNA. They're haploid by chromosomes, but they're not haploid by DNA content, because haploidism, or diploid, haploid refers to chromosome number, not necessarily DNA content. Then these are called secondary spermatocytes. And then the secondary spermatocytes then each undergoes the second meiotic division. I'll put this board down in a second here. And these guys form 4 spermatids. And the spermatids all are true haploids. So the spermatids are 23 chromosomes and cDNA. So it's a true haploid. Okay? Then each of these spermatids differentiate into spermatozoa. Which are also, obviously, these are the true haploids also, because they're 23 chromosomes and cDNA. And this is the gamete. So from each spermatogonia, how many spermatozoa do you end up with in males? 4. Good. Keep that in mind, because females are completely different in that point of view. Not so much in the spermatozoa side, but in forming haploid gametes. And it's a continuous process. Okay? And the whole idea here is to get this haploid gamete. And this is what goes on in the seminiferous tubules. Okay? Any questions? Are we good there? All right. So you make this spermatozoa. And just to give you a sense of the structure, what gets produced in the seminiferous tubules is not very mature, but we'll get to that in a second. But the spermatozoa has a very straightforward structure. There's an acrosome, which has enzymes for digesting the egg membrane. And to use the word egg for females very generically, digestion or hydrolysis of it. There's the nucleus. And what's the DNA content of the spermatozoa nucleus? Haploid, right? 23 chromosomes, cDNA. There's a bunch of mitochondria. For energy. And then there's a tail for motility. Okay, so it's basically it moves around with one purpose, and that's to crash into the egg, digest the membrane, and shoot the haploid nucleus into the egg. That's the entire role of this thing in terms of what it does. Okay. Now, to get to a spermatozoa that actually works, there's a maturation process. And we'll briefly talk about that before we talk about the male hormones. So you start out in the seminiferous tubules. And what's the seminiferous tubules look like? How did I describe it on Monday? Like a big ball of spaghetti, right? Or a big plate of spaghetti, I should say. And there's many ducts. And the spermatozoa that you produce there are not mature enough to function. And over the next 20 or so days, approximately 20 days, the spermatozoa mature. So, and basically, so they're more mobile, essentially what's going on. And they all go into a single duct called the epididymis, which is a single duct with mature spermatozoa. Okay? One form of male reproductive problems, by the way, is the inability to properly mature the spermatozoa, so they don't move around well enough. And that somehow takes place in this process here. Part of the reason. It's one reason. Another reason, obviously, is not producing enough spermatozoa. But you can produce spermatozoa that are not mature. So the epididymis, I'll just continue with that, it goes into the vas deferens structure. And then the vas deferens goes into what's called the ejaculatory duct. Now what's important here is there's several glands produce material that go into this. And these glands are the seminal vesicles. You don't really need to distinguish what each one does. You have the prostate gland. By the way, it's the prostate gland that's highly testosterone sensitive. And for men, if you live long enough, you're always going to get cancer of the prostate gland. So this is a very big issue as males get older. But it's, for whatever reason, it's the prostate gland that's affected most in this entire system. You also have the bulbourethral gland. And you don't really need to distinguish these, but together, these guys provide sugars, certain hormones. They provide sugars, hormones, some enzymes and protein, and buffering capacity. Primarily sodium bicarbonate. But you don't need to know that. You just need to know in total, these glands produce fluid, hormones, enzymes, and so forth. And then from there, you have the ejaculation, and you produce in the neighborhood of 100 to 300 spermatozoa, 100 to 300 million spermatozoa. Again, you should just say, as we talk about the females, you need to see the fact that males are producing hundreds of millions of gametes, and the female maybe is producing 1 or 2. Okay, so there's a very big difference reproductively. Okay? Any questions on this pathway? The first part's spermatozoa maturation, and then you get it all ready to deliver, and it's done by several glands that feed into the ejaculatory duct, and you produce the spermatozoa. Okay. So and I'll bring this board back up here in a second. Again, in males, one of the big issues is erection. And basically, it's a signal transduction process, a hormone-like signal transduction process. Through nerve impulses, primarily, but there's others. This is sort of the simplified version. You produce a gas called nitric oxide. This doesn't mean just say no, it's nitric oxide. And nitric oxide is a hormone-like in its action. And I say that, it's clearly not a hormone, but what it does do, something we talked about on Monday, it activates through several steps a guanylate cyclase, which converts GTP to cyclic GMP, and cyclic GMP causes smooth muscle relaxation. And you don't need to know the mechanism, but there's some phosphorylation events, ion channel changes, and so forth. And it causes arterial dilation. This is what causes the erection. And as we talked about is cyclic GMP is broken down by a phosphodiesterase. Again, we talked about this Monday. And I'm writing it small here. The 5' GMP. And Viagra inhibits this phosphodiesterase. So you keep cyclic GMP around much longer, much more extended times. Okay, in fact, in the commercials, call a doctor if you stay erect for over 4 hours or something like that, right? And it's because you're keeping cyclic GMP around much longer than you physiologically normally can do, and induces this process. Okay, as we talked about, also that same phosphodiesterase is in the rod cells in your eyes. And so one side effect for about 10-20% of people who take Viagra is temporary blindness or temporary visual problems. Okay? Questions on that? So basically, so that's the basic process, working in a hormone-like signal transduction pathway. So you can bring that out in conversation next time you decide to talk about this pathway. Okay? All right. Male hormones. Males do this, have a continuous process due to negative feedback. So again, we're going to talk about the hypothalamus, anterior pituitary axis. The hypothalamus produces a hormone called GnRH, gonadotropin releasing hormone. H is hormone here. And this binds to, how does every self-respecting hormone work? What must it interact with to be called a hormone? A receptor. Very good. Binds to a cell surface receptor. I'll put this board up here in a second here. In the anterior pituitary. Okay. And the activation of this receptor in the anterior pituitary stimulates several hormones. Now, the hormones in males are the same as females, and therefore has maintained the same names. So one of the hormones that gets produced is luteinizing hormone, or LH. Obviously, when we talk about female reproductive system, a surge in LH is very important for ovulation. It was discovered in females first, same hormones in males, still called LH. It produces another hormone. There's other hormones involved, but these are the main ones, called FSH, follicle stimulating hormone. Again, males do not have follicles, but they have the same hormone. Okay? Luteinizing hormone, being a hormone, works through a receptor. Its most important receptor is in the Leydig cells. And this activates the conversion of cholesterol into testosterone. So it activates a signaling pathway. This is multiple steps. Between cholesterol and testosterone is about 6, 7, or 8 steps. It activates that whole pathway. FSH, through its receptor in the Sertoli cells, causes the conversion of testosterone into dihydrotestosterone. And it is dihydrotestosterone that keep the spermatogenic cells going. Activates the meiotic process, mitosis, and so forth. So it is not testosterone that does it. Testosterone is merely a precursor for dihydrotestosterone. Okay? Any questions at that point? And then testosterone besides going between the Leydig and the Sertoli cells, also goes out into the system. Okay, gives you a lot of your, maintains male characteristics. Now, what the other thing testosterone does, and I'm going to sort of give you a smaller version of this. This is the hypothalamus. This is the anterior pituitary. And this is the Leydig and Sertoli cells. So GnRH activates the anterior pituitary, which activates, in this case, LH and FSH in the Leydig and the Sertoli cells. This testosterone that comes out of the Leydig cells, besides having these systemic effects, also provide a negative feedback. There are receptors for testosterone in the hypothalamus and in the anterior pituitary. So it provides a classic negative feedback system. You produce GnRH, you produce LH and FSH, you make testosterone for conversion into dihydrotestosterone. A lot of the testosterone goes out into the system. When there's too much testosterone, you then turn off LH and FSH production, and you turn off GnRH production. When that gets too low, you go below a threshold, and then you kick up the synthesis of GnRH and production of GnRH and LH and FSH and make more testosterone. So testosterone's low, you make more. Testosterone's too high, you make less. Question in the back. I'm not sure why it's kept the temperature relationship. It has probably something to do with the maturation process, because in the epididymis you also store spermatozoa for a long time also. It's not just you're there and every time you have enough you ejaculate, right? Sometimes it can be stored for fairly long time frames. Okay? So I'm not 100% sure how to answer your question, but I'll look it up. What the relationship of temperature is to that. But it probably has to do with maturation and viability, is my guess. Good question. Okay, so you have this classic negative feedback system. You have too much testosterone, you turn it down, you modulate it down. You have too little, you modulate it up. Now, given this, if you have a negative feedback system, so you maintain this level, if you give the system too much of the negative regulator, you can dampen if you give it not physiologically, but you give it as a medicine or a drug, you can bring down the entire system if you provide this negative regulator. So given this, what would make a good male contraceptive based on this discussion? Just based on what I just said. What brings down the negative feedback pathway? Extra what? Testosterone, exactly. So you look at this and you say, some company looks at this and says, okay, we just have to give a male extra testosterone and we're going to dampen this system. And it's exactly what happens. However, there are side effects of giving a male a lot of extra testosterone. Everything from physiological to behavioral disorders. Okay? Well, what they found is, surprisingly, that testosterone plus progesterone, if you give that, it dampens this pathway because you maintain the negative feedback. And let me just put this up here. And importantly, you knock down dihydrotestosterone production, right, by dampening this whole system. But by adding progesterone, for reasons that's not clearly understood, at least to me anyway, that you dampen the systemic effects of extra testosterone. So you get rid of the negative part of this thing, the difficult part of this thing, physiologically and behaviorally, by giving a little bit of progesterone. And this is what they're working on right now, is a sort of an alternative to vasectomies for males. Okay? And some of you, later on, as you become doctors, might be prescribing versions of this. Okay, any questions? Okay, so keep in mind, with males, it's a continuous process due to the negative feedback. You also make lots and lots and lots of gametes. Okay? Females have a very different scenario. Get another piece of chalk here. And in females, I guess we'll do this over here. In females, The whole system is called oogenesis. And this is ovum production, which is the gamete, or as you'll see, there's not really a cell that's simply an ovum. It's really an ovum nucleus. And we'll give a generic term, call this eggs, because there are different stages of the oogenic pathway that have very unique characteristics. So what are some of the key issues of oogenesis? Okay, for one thing, unlike males that are continually making haploid gametes, females, for the most part, are born with all their needed, we'll call these eggs for now, we'll give it a name in a few minutes. They're born with all their eggs. And we'll just look at a developmental pathway here. Here's the eggs in quotation marks. This is conception. And the female is typically born, this is when they're born, with about 1 million eggs. And by puberty, what you have left is about 400,000, which, by the way, is way more than you'll ever need. So you do have extra, many, many extra eggs. This is unlike males that are continually producing it. You also have long resting periods in the oogenic pathway. So for a female that ovulates at the age of 50, that particular egg's been sitting there for 50 plus years before it continues in the process. Okay? For males, that would not happen. Secondly, another part of this is, and you'll see why this is in a minute, there's unequal cytokinesis, which is the cytoplasm, division of the cytoplasm. Okay? And that's also a big difference with males. Another part of this is the object of a female, obviously, is to make a gamete. However, a female only makes a potential, I stress the word here, potential ovum approximately every 28 days. And those of you can recognize the time frame of the menstrual cycle. Most women in their lifetime maybe make 5 to 10 true ovums, ovum nuclei, in their lifetime, maybe. And you'll see why that is. They make a lot of potential ovums every 28 days, but not necessarily ovum. And finally, this all takes place in the follicle of the ovary, which has call this oogenic cells. And there are accessory cells. As you might predict, you've got theca cells, which converts cholesterol into testosterone. Note, females make testosterone. But then you have the granulosa cells that converts testosterone into estrogen through an important enzyme called aromatase. Okay? So, if I say to you testosterone, truly you cannot say it's only produced in males. Okay, it has the biological effects in males because we produce the receptors for androgens, but it is absolutely critical and produced in females. Questions on that? Yes?
Is all the testosterone?
Yeah, what's the efficiency of conversion to estrogen? Nearly complete. You can have defects in aromatase that change reproductive efficiency. Secondly, because a lot of breast cancers, certain kinds of reproductive cancers are hormone responsive, some of the new wave drugs that have been developed in recent years is to inhibit aromatase as a way of dampening the estrogen levels, and therefore as a potential treatment for breast cancer. And actually, it looks like it's a little more effective right now for a lot of patients, certain patients, than tamoxifen, which is an anti-estrogen. It's an estrogen receptor antagonist. So this enzyme is very important. People are paying a lot of attention to it. Yes, question?
Is aromatase present in males?
No. Is aromatase present in males? Very, very little of it. Males make a little bit of estrogen, and it's important for bone development. Very little. Very little. So again, to say that males don't produce any estrogen is also not true. It all comes down to the receptors, by the way. Who has the receptors? And which tissues and which amounts and so forth. It is not necessarily the hormones. Okay? So kind of get around saying estrogen is the female hormone and testosterone is the male hormone. It's not true. What you could say perhaps is dihydrotestosterone is the male hormone, but not testosterone. Females do not make dihydrotestosterone. Okay, questions? So in this oogenic pathway, and we'll get to these key differences to the male, between male and female here. So in females, as I said, you're born with about 1 million of these eggs. What happens is, during fetal development, there's something called primordial stem cells, which undergo mitosis and differentiation, form oogonia. And both the stem cells and the oogonia are 46 chromosomes and 2 cDNA. So these are true diploids. At some point between conception and birth, at some point, the oogonia undergo the meiotic duplication of the DNA, forming primary oocytes. And these primary oocytes have 46 chromosomes but 4 cDNA. This is what the females are born with. Okay? So we're calling them eggs here, but the reality is what's born in through puberty are primary oocytes. So females are born with a potential ovum that's in the 46 chromosome 4C DNA content. And they just hang around there, waiting for some important events to continue the process. And what happens is, right during ovulation, which we'll talk a little more detail about, you've got a surge in LH, luteinizing hormone, and you have ovulation, and you have the first meiotic division. And you have unequal cytokinesis. So what happens is you form a secondary oocyte that is 23 chromosomes and 2 cDNA, and it has virtually all of the cytoplasm. In addition, because you're splitting off the rest of the DNA, you get a small little thing called a polar body, which is a 23 2C nucleus, but virtually little to no cytoplasm. Okay? And this basically gets eliminated. Okay? Everybody see the movie Twins with Danny DeVito and Schwarzenegger a long time ago? Well, the theory behind that whole movie is Schwarzenegger got the secondary oocyte, kept going, and DeVito got the polar body. Okay? That's sort of like the underlying sci-fi part of that movie. Yes?
So is this what decreases it from like 1 million to like 400,000, or is it because it's
Oh, no. During puberty, which is when ovulation can take place, you're at 400,000. Between here and here, you lose about half of them. They get digested. That'll become more clear when we talk about follicle development. Okay? So at this point, you have a secondary oocyte. And this is what gets ovulated. This is what gets kicked out of the ovary. This is what I call the potential ovum. It doesn't always go into an ovum, but it has the potential. Okay? So let's go over here. And how does that form an ovum? That begs that question. So you have the secondary oocyte. This does not form an ovum nucleus until you have fertilization. Okay? So what happens is you've got fertilization. Actually, this all takes place in the fallopian tubes, which we'll talk about Friday. And what I call fertilization is, my definition is, entry of the spermatozoa nucleus. So you basically have a cell at this point that has the secondary oocyte nucleus and the spermatozoa nucleus. And as a result of the entry of the spermatozoa, there's massive changes in metabolism in the cell, and you get the second meiotic division. And what you get is another polar body and the ovum nucleus, which is 23 chromosomes and cDNA. So this turns into the ovum nucleus. So at no time do you have an ovum cell that's only the nucleus. What you have is a cell that has two haploid nuclei. So you never get it with just the ovum nucleus. It has to have the spermatozoa nucleus in there. Okay? And then what happens is the nuclear membranes break down. The DNA recombines with the DNA, comes back together, and you form your zygote, which is obviously diploid. So at no time do you have a separate ovum nucleus in there. So that is why in female reproduction, you have a potential ovum produced every 28 days. You need fertilization to get that final gamete haploid nucleus. Does everybody see that? Okay? So that is why I say most females maybe produce 5 to 10 ovum nuclei in their lifetime, or less. But about every 28 days, you produce a secondary oocyte. Question?
Over here you have the oogonium going to the primary oocyte, and you say it's mitotic amplification.
Yeah, that's duplication. Mitotic amplification is duplication. I call that duplication of the DNA to impress upon you that you're doubling the DNA. Well, either way, because it leads to meiosis. I will change that for next year. Thank you. It's DNA duplication, which then leads to meiosis. It does not lead to mitosis. Yes?
Where does the secondary oocyte's nucleus come from when it's all cytoplasm?
The secondary oocyte is what a female ovulates. It comes from the primary oocyte. The primary oocyte DNA divides. You've got a 23 chromosome 2C nucleus that becomes the oocyte, and you have a polar body 23 2C nucleus that does no cytoplasm, and it gets dissipated, digested. So from any one oogonia, how many ovum nuclei can potentially be made? One. From one spermatogonia, how many spermatocytes can be made? There you go. You guys have it. How many polar bodies can be made from one oogonia? Well, it's usually 2. Usually. There's one of these polar bodies can also divide, but it doesn't happen all that often. It's usually 2 because it's a dead-end process. Right? You get a polar body from the primary oocyte, and you get a polar body from the secondary oocyte. Sometimes they divide, but not always. I wouldn't ask you that as a question, by the way. All right? Questions on that? Yes?
So, if we have like 400,000 potential oocytes in the ovary, like how does the body choose which one?
Very good question. That's the question is, how do you choose which of the 400,000 to continue? with the process. If you think about it, one primary oocyte develops every 28 days. That's about 12 to 13 a year. You're reproductively efficient between 20 and about 50. So 30 years times 20, you're only talking about 300 or 400 total out of 400,000. And that's the perfect segue, thank you for the question, into the next topic, and that is follicle development. The short answer is nobody knows that answer. The short answer. And that is because, as I said, it all takes place in the ovary. Here's the ovary. And you have 400,000 follicles. Well, at the start of the menstrual cycle, about 20 or so in each of these follicles are called primary follicles. They're called primary follicles because they have a primary oocyte sitting in there. So you have 400,000 of these follicles in the ovary. At the beginning of the menstrual cycle, about 20 or so of these follicles start developing. And this is due to the actions of gonadotropin, LH, and FSH, which we'll get to. About 20 start developing. For reasons nobody understands, one of them tends to win out and becomes the primary follicle for the rest of the development. Nobody understands that. As you can imagine, sometimes two develop. And guess what happens when two develops? Twins, right. And I have a set of twins, and that's likely what happened. Okay? So and also, fertility drugs, one class of them we'll talk about on Friday briefly, causes multiple follicles to develop, but you don't have the off mechanism, whatever that is. So women on fertility drugs can have multiple oocytes develop. And so what's the side effect of fertility drugs you guys read about? Triplets, sextuplets, 8 octuplets, whatever they are, you know. Multiple births, because you can't stop this process, this natural process. So it's you have to fine-tune how much you can take to do that. Okay, does everybody see that? But the bottom line is people do not know which of these becomes the primary follicle for development. Now what happens is so once that occurs, once it is chosen, and also, by the way, genetically, some women are always developing two or three at a time. And that is why in families, you can see twins will occur in families. Because for reasons people do not understand, some people have a predisposition for producing more of these follicles. So you have proliferation. What I'll end with is, what happens is you have a primary oocyte inside of a membrane. And this membrane is called the zona pellucida. This is a very important membrane. It has spermatozoa receptors. And outside this is a layer of granulosa cells. What do granulosa cells make from testosterone? What do they make from testosterone? With aromatase. Estrogen, right. And outside the granulosa cells, you have a layer of theca cells, which produces what? Cholesterol into what? To testosterone. So in theca cells, you go cholesterol into testosterone. We'll talk about it a little bit later. And granulosa cells goes testosterone into estrogen. The estrogen then goes into the primary follicle and helps with the growth. This is very important to realize. As the cells divide, they produce more estrogen at a certain point. And that'll be where we'll pick up on Friday, because we'll get into the menstrual cycle, female hormones, and then pregnancy. Okay? Any questions? We have like 30 seconds here. Any questions? Okay, again, think about phosphodiesterases and signal transduction pathways and hormonal control mechanisms, and you have all of human reproduction down. Okay, see you Friday.